Welcome to this video. In this video we will see plant regeneration methods, somatic embryogenesis. First we will see the difference between embryogenesis and somatic embryogenesis. Embryo is the plant in its initial stage of development and it has two distinct poles, one to form the root and another one to form the shoot. In some plant species, embryos are produced without the fusion of gametes and called as asexual embryogenesis or adventitious embryony. These embryos may occur in sporophytic tissues like integuments or nuclear tissues from unfertilized gametic cells. Apart from this zygotic embryo and asexual embryogenesis, embryo formations from tissue culture in vitro were reported, and this phenomenon is termed as somatic embryogenesis. The scientist Stewart in 1958, Reinhardt in 1959 first reported the somatic embryogenesis in vitro culture. Let's see difference between somatic embryo and zygotic embryo. Somatic embryos are formed from ordinary plant tissues, mostly like for example diploid plant tissues that are not usually involved in the development of embryo. That means they are not gametic cells. No sexual embryo and endosperm and seed coat is formed in the case of somatic embryo. Somatic embryogenesis is the process where a plant embryo is developed from somatic cells. So in the following picture shows the difference between somatic embryogenesis and zygotic embryo. So in somatic embryogenesis, callus is formed. From the callus, globular cells are formed. From there, heart-like shaped cells are formed and further differentiated to torpedo or torpedoid-like cells. Then it is matured into miniature plantlet with bipolar. Whereas in the case of zygotic embryo, zygote will form after the fertilization of gametes, then it undergoes globular stage, heart stage, torpedo stage and cotyledon or seed will form. Somatic and zygotic embryos are most similar from the globular stage through the torpedo stage. But after maturation, unlike zygotic seed, somatic embryo do not experience desiccation or dormancy and continue to grow in fully differentiated plantlets. Let's see the pathway of somatic embryogenesis. So mother plant is selected, then explant is excised and culture in vitro medium suitably trigger the embryogenesis process. Step 1 callus will occur, next embryoid will occur. The embryoid is generally used to denote the embryo-like structure from cultured tissues. And these embryoids possess bipolarity, no vascular connection with the mother tissue and origin from a single cell or a group of cells. From embryoid, further it is matured to somatic embryo. Then each somatic embryo can capable to produce one plant. Since it have bipolar and carrying future shoot and root cells, there is no necessary to modulate or induce the shoot or root with different growth hormones like auxins and cytokines. There are 6 theories attempted to explain the process how somatic embryogenesis takes place. First theory is cell isolation theory proposed by Stewart in 1964. So he told the embryo producing cells are physically or physiologically isolated due to constraints in the surrounding cells and they are forced to form embryo. Next theory is differentiation theory proposed by Halperin in 1970. He told it is a prerequisite for the production of somatic embryos in vitro in some cells. And third theory is intercellular communication and cytodifferentiation theory. Street is proposed this one. Cytodifferentiation by diffusion gradients of nutrients, endogenous growth regulators and gaseous factors like oxygen, CO2 induces embryo formation is explained by this theory. Fourth theory is explant physiology and culture environment theory. Street 1978 proposed this one. The embryogenesis is dependent on the phenomenon of explant and cultural environment condition. Fifth theory is predetermination theory proposed by Tissarat et al. Embryo production potential is predetermined phenomenon in the cells, and in vitro culture provides the opportunity for the formation of embryo. And last theory is pre and induced embryogenic determined cell theory. Sharp et al. Proposed this. Through the embryogenesis is predetermined, there are instances of non-formation of embryos directly from the explants. Two types of cells are proposed in this theory. One is pre-embryogenic determined cells and induced embryogenic determined cells. Generally, somatic embryogenesis formed by two patterns. One is called direct embryogenesis, another one is called indirect embryogenesis. In direct embryogenesis, embryos form from the explants directly. In indirect embryogenesis, embryos arise from the callus induced from the explants. In direct embryogenesis, a promoting substances to induce the embryo formation is needed, whereas mostly in the indirect embryogenesis, auxin is needed to induce the callus and cytokinin is needed to induce the differentiation. In direct embryogenesis, the embryogenic nature of a cell is predetermined, whereas in indeterminate embryogenesis, the embryogenic nature of a cell is induced in the culture itself. In direct embryogenesis, the origin of embryos mostly from individual cells, sometimes or rarely from a group of cells. Whereas in indirect embryogenesis, the origin may be either from single cells or from group of cells called proembryonal complex. So let's see the applications of somatic embryogenesis. First application is clonal propagation of genetically similar plant material is possible, and second application is elimination of disease such as virus and pathogens is possible through this method, and third one is providing source of tissues for genetic transformation experiments, and fourth application is production of whole plants from single cells known as protoplast. The fifth application is development of synthetic seed technologies. So since it is very small in size, we can easily develop synthetic seeds in large number in small area. Next we will see some of the problems associated with somatic embryogenesis. So generally it have 6 major problems. One is high chance of mutations. Two is the method is little bit difficult, need more skill and experience. And third one is loss of ability to regenerate or reproduce in several cases. And somatic embryogenesis results in high percentage of albino shoots during regeneration, and the somatic embryogenesis process is not possible with all the plant species. Only those plants responsive to this kind of differentiation can successfully produce the somatic embryo. And finally, somatic embryogenesis methodology must be optimized for each species. Even within the species, variety to variety or genotype to genotype, we have to optimize. So thank you.